A recent multi-cohort study of over 8,000 people found that socially isolated individuals carry a 24% higher level of a chronic inflammation biomarker called SUPAR. This biological shift makes loneliness as dangerous to your cellular health as smoking 15 cigarettes a day. We often treat aging as a simple ticking clock, an inevitable rusting of the machine. But from a biological standpoint, aging is deeply tied to a phenomenon called inflammaging. It is a persistent, low-grade systemic inflammation that slowly degrades our tissues over decades. The medical consensus has long pointed to poor diet, obesity, and even dental health, like periodontal disease, as the primary drivers of this chronic inflammation. We are constantly told to eat antioxidant-rich foods and floss our teeth to protect our hearts. But the scope of what actually inflames us is much wider and, frankly, much more integrated into our modern lifestyle. We are looking at environmental and psychosocial triggers today that fly under the radar. Before we get into things like microplastics or light pollution, I want to clarify this concept of inflammaging. How does this low-grade fire differ from the inflammation we experience when we twist an ankle? The distinction is fundamental. Acute inflammation is a life-saving evolutionary mechanism. If you twist your ankle or get a bacterial infection, your immune system dispatches white blood cells, specifically macrophages and neutrophils, to the site. The area gets red, hot, swollen, and painful. That is the immune system clearing debris and initiating repair. Once the threat is gone, the inflammatory response shuts down. Inflammaging, on the other hand, is a chronic, sterile inflammation. There is no acute infection and no twisted ankle. Instead, your immune system receives a constant, low-level signal that something is wrong. Because the signal never stops, the immune cells never stand down. They continuously secrete inflammatory chemicals called cytokines, like interleukin-6 and tumor necrosis factor alpha. Over time, these chemicals damage healthy cells, mutate DNA, and create fibrotic, stiff tissue. This process is now recognized by geroscience as the root driver of cardiovascular disease, Alzheimer's, and metabolic syndrome. Let's look back at the statistic I mentioned at the start. I think most people accept that physical substances, like a diet high in refined sugars, can trigger a physical immune response. But the idea that a purely psychological state, like loneliness, can elevate a physical biomarker like SUPAR requires a leap. It suggests our immune system is actively listening to our social lives. How does an emotion translate into a cellular inflammatory response? It requires looking at human biology through an evolutionary lens. For early humans, social isolation was not just sad, it was a physical death sentence. If you were separated from your tribe, you were highly likely to be attacked by a predator or a rival group. Consequently, human biology evolved a brilliant, if now problematic, survival mechanism. When the brain perceives social isolation, it activates what geneticists call the conserved transcriptional response to adversity, or CTRA. The nervous system signals the bone marrow to alter gene expression. It aggressively upregulates genes that promote inflammation, anticipating physical wounds, while simultaneously downregulating genes responsible for fighting viral infections. Your body literally prepares to be torn open because it knows you are alone. In other words, your immune system is making a calculated bet. It sacrifices viral defense to flood the body with inflammatory agents, assuming a physical attack is imminent. But in the modern world, the isolated person is not wandering the savannah facing predators. They are sitting in an apartment, completely safe from physical trauma, yet their immune system is locked in this state of high alert. The mismatch between our evolutionary programming and our modern environment is where the damage occurs. According to data published in Brain Behavior and Immunity, this chronic stress response dysregulates the hypothalamic pituitary adrenal axis, leading to persistently high cortisol levels. Eventually, the cortisol receptors become resistant, and the inflammatory cytokines run wild. This is why the research shows that isolated individuals experience accelerated telomere shortening, which is the biological marker of cellular aging. The psychological pain of loneliness is processed by the same neural circuits as physical pain, and the immune system reacts accordingly. It is a fascinating but tragic example of an ancient survival mechanism accelerating our demise in a modern context. It is a profound reframing of how we view public health. We cannot just prescribe a Mediterranean diet to someone whose immune system is actively preparing for a life-or-death physical trauma due to isolation. But I want to pivot from the psychosocial to the deeply physical, specifically the environmental toxins we are saturated in. There is growing scrutiny on microplastics. We know they are in our water, our food, and even the air. I have read they have been found in human blood, but we need to understand their role in this inflammaging process. Do they just pass through us, or do they actively trigger an immune response? The answer is that they absolutely trigger an immune response, and the clinical data emerging right now is staggering. A landmark study recently published in the New England Journal of Medicine examined patients undergoing surgery for carotid artery disease. The researchers found microplastics and nanoplastics, specifically polyethylene and polyvinyl chloride, embedded directly within the arterial plaque of a massive percentage of the patients. Those patients with plastic in their plaque were over 4 times more likely to suffer a heart attack, a stroke, or die during the follow-up period compared to those without detectable plastic. That correlation is terrifying. But mechanistically, what is the plastic doing inside the artery? Is the plaque formation a direct immune response to the foreign particle? The answer is yes, it acts as a permanent irritant. When a microplastic particle embeds in a tissue wall, whether it is a blood vessel or the gut lining, the immune system dispatches macrophages to clear it out. The macrophage attempts to engulf the particle, a process called phagocytosis. But because the plastic is synthetic and cannot be enzymatically broken down by the cell, the macrophage essentially chokes on it. The immune cell dies, releasing its toxic internal contents and a flood of inflammatory cytokines into the surrounding tissue. This calls more immune cells to the area, which also fail to digest the plastic, creating a localized, never-ending loop of inflammation and cell death. This necrotic core is what destabilizes arterial plaque, making it prone to rupture. It seems like that our body is essentially fighting a war it can never win, and the collateral damage is our cardiovascular health. But it is not just the arteries. I was looking at a 2024 study in Scientific Reports detailing how microplastics accumulate in adipose tissue, our fat cells. We typically view fat just as energy storage, but adipose tissue is actually a highly active endocrine organ. Adipose tissue is heavily involved in immune regulation. The study found that when microplastics accumulate in white adipose tissue, they induce cellular senescence. Senescent cells are often called zombie cells. They stop dividing, but refuse to die. Instead, they secrete a highly toxic cocktail of inflammatory markers, known as the senescence-associated secretory phenotype, or SASP. The microplastics essentially force the fat cells to age prematurely and malfunction. They also impair adipogenesis, meaning the tissue loses its ability to store lipids properly, which forces those lipids into other organs like the liver and heart. This single mechanism ties microplastic exposure directly to insulin resistance, systemic inflammation, and metabolic syndrome. That provides a much more nuanced picture of obesity and metabolic disease. It challenges the assumption that metabolic dysfunction is purely a failure of willpower or caloric balance. If environmental pollutants are artificially aging our fat cells and triggering chronic inflammation, we are dealing with a systemic toxicity problem. And yet, there are even less tangible pollutants driving this process. We have talked about emotional isolation and physical microplastics. But what about environmental factors we cannot even hold? For example, the light and noise pollution. We treat ambient city noise and artificial lighting as mere nuisances, but biological data suggests they are doing real damage. Let us start with artificial light at night, often abbreviated as ALAN in the literature. For billions of years, life on Earth evolved under bright days and profoundly dark nights. Our circadian clock, which exists in virtually every cell of our body, is calibrated by this light-dark cycle. When we expose ourselves to artificial light after sunset, especially the blue wavelengths emitted by screens and LED streetlights, we send a false daytime signal to the suprachiasmatic nucleus in the brain. This immediately halts the production of melatonin. Most people view melatonin simply as an over-the-counter sleep aid, a hormone that makes you drowsy. But its role in the body is much broader than that. Calling melatonin a sleep aid drastically undersells its biological importance. Melatonin is one of the most powerful systemic antioxidants and anti-inflammatory molecules the human body produces. It actively scavenges free radicals and modulates the immune system. When light pollution suppresses your melatonin, you are not just losing sleep, you are stripping your body of its primary defense against daily cellular damage. Studies published in the Environmental Health Perspectives have linked high exposure to artificial light at night with significant increases in breast and prostate cancers, largely due to this disruption of circadian rhythm and the resulting chronic inflammation. It seems the loss of the dark is a massive physiological stressor, and chronic noise acts along a similar pathway. A siren outside your window might not wake you up fully, but your brain still registers the acoustic intrusion. Our auditory system never sleeps. It evolved as an alarm system to detect approaching threats. When you live in an environment with chronic noise pollution, such as near a busy highway or an airport, your brain registers those sounds as danger signals, even if you are consciously habituated to them. This triggers the amygdala, which activates the sympathetic nervous system and the HPA axis. Cortisol and adrenaline are released into the bloodstream. This sustained elevation of stress hormones damages the endothelial cells lining your blood vessels, promoting oxidative stress and the buildup of plaque. The World Health Organization has explicitly recognized environmental noise as a major driver of cardiovascular disease precisely because it sustains that low-grade inflammatory state. We are uncovering a deeply interconnected web here. Whether it is the psychological strain of loneliness, the physical intrusion of microplastics, or the sensory assault of artificial light and noise, the biological outcome is identical. The body perceives a constant threat, locks the immune system in a constant active state. That is the defining paradox of modern civilization. We have largely eradicated the acute threats that historically killed humans, like predators, starvation, and acute infectious diseases. But we have replaced them with a pervasive low-level environmental hostility. The conveniences of modernity, the synthetic materials, the illuminated cities, the shift away from multi-generational community living are interpreted by our ancient biology as a continuous state of emergency. Inflammaging is simply the biological cost of this mismatch. It forces us to rethink how we approach aging. It is not just about what we eat or how often we visit the dentist. It requires actively curating our environment to signal safety to our biology. That means prioritizing darkness at night, cultivating genuine social ties to turn off the CTRA response, and minimizing our exposure to synthetic materials wherever possible. We have to give our immune system permission to rest. If you found this video valuable, send it to a friend who might benefit from it.